Hello, and hopefully the tech is working. Welcome to Science Dreams again. I know we got a couple of viewers here already. Thanks for stopping in and being here. And with us is Ryan Haupt, which is just awesome to have you here with us today. And just go ahead and tell us about yourself. I do have the slide up, the first one of your dissertation.
Yeah, so my name is Ryan Haupt. I just finished my doctorate in Sloth Ecology and Paleoecology at the University of Wyoming, and I'm currently living in DC. So, and I'm a big fan of comics, as I think we're going to get into as we talk during the stream, but I'm just excited to be here and happy to talk about all things sloths and science and comics.
So I was kind of worried at first. I'm like, all right, so we're going to probo this sloths, known for being really slow and I don't think boring, but some people might, and evolution, which is also very slow. This is going to be a tough sell maybe. But then I realized how much of pop culture loves sloths. I mean, they have the natural smile, right? So
the three-fingered ones do. The two-fingered ones have more of a piggy snout, which is cute in its own way, but it's not the perma smile of the three-fingered ones.
Right. What got you interested in sloths to begin with? What was the main thing that really sold you on them?
That's a great question. I think most people think that I came to it because I was sort of obsessed with them, but really I started my master's. I went to the University of California, Santa Cruz for undergrad, and I ended up studying ecology and evolution, but I also ended up studying paleontology, and so I ended up double majoring with a geology degree as well. And then when I went to Vanderbilt for my master's, I worked with my advisor, Dr. Larissa DeSantis, and we were sort of just discussing potential master's projects one day, and she was getting into the idea of this technique called dental microwear texture analysis, where you look at the microscopic surface textures of teeth as a sort of indicator of what the sort of food that the animal was chewing on, or the structural properties of that food. And she didn't know if this technique would work on animals like sloths and armadillos because they have relatively soft teeth compared to most other mammals. We all have enamel on our teeth, which is pretty common for mammals. Sloths and armadillos, which are closely related to sloths, have lost the enamel and just have dentin teeth, so their teeth are just physically softer than typical mammal teeth, and she wasn't sure if this technique would work. Are the teeth too soft to record this information? And so she kind of was just like, why don't you give it a try? And I started digging in, and the more I learned about sloths and armadillos too, but sloths in particular, the more I was like, wow, these are just really weird animals, and frankly there's a lot of kind of basic information we don't really know about them, even though they have this really amazing fossil record and evolutionary history that I found really fascinating.
Cool. Hey, Uncle Bill, welcome into the channel. Good to have you here. And you just caught us at the beginning of talking about sloths. Hey, Outlier, thanks for the sub. I appreciate that. Awesome to have you here. Please, if you have questions about sloths, this is the guy. There's not many experts on sloths. I'm sure there are, as far as like zoo handlers and people that maybe work on ecology, but it's the evolutionary history. How many people can there possibly be that is their specialty?
I'm on a first name basis with most of them, I would say. There's sort of there's maybe a dozen or so people, maybe more, maybe 24 if we're really stretching it. But yeah, it's a relatively small community, but we all mostly get along and have a good time. I would say it's, there's a sort of paleo group and then more of a modern biology group, and there's not a lot of people that work at sort of the intersection, which is one of the spaces where I think I have a lot of fun and think about things maybe a little differently than a lot of my fellow slothers out there.
Slother. So when you go to the sloth convention, is there a particular debatable topic? And like you get into it with them and you're like, hey, no, that's not the way it works.
There's, I would say, less debates and more just kind of open questions. Like, it's still a pretty open question how they ended up in the trees. we have this amazing fossil record of all these ground sloths, but within and when I say amazing fossil record, I mean we have a fossil record going back almost 40 million years of ground sloths in South America. They started in South America. They very quickly distributed all throughout South America. They crossed into Central America and North America before the Isthmus of Panama finished forming. So this was marine dispersal, these sloths swimming across shallow seaways. And they distributed all throughout the Caribbean as well. They made it as far north as the Yukon Territory in Canada and parts of Alaska. We find them on the tops of mountains in Colorado. We find them coast to coast in what is now the U.S. The tar pits of California and cave sites in here on the East Coast and places down in Florida. So really wide distribution, 80 to 90 genera among 5 or 6 different families. And today there's only 2. There's only 2 kinds of sloths, about 6 species. So we're living in a world that's lost most of its sloths. And
does that make you sad?
What was that? Oh, yeah, it does make me sad. It totally makes me sad. Especially considering how recent some of them went extinct. The last surviving ground sloths were walking around 4,500 years ago, which is like nothing in deep time. The Great Pyramids of Giza had been built by then. So that's the world that the sloths went extinct from. And we're lucky we still have 2, but it would have been cool to have more. And to answer the question you asked had to do with the questions that we still have. And the question that one of the questions that I think about a lot is all the fossils we have are ground sloths. We have a few fossil sloths that maybe could climb trees if they wanted to, but they could definitely get around on land if they wanted. And so this idea of these sloths being this exclusively tree-dwelling animal today kind of comes out of nowhere, evolutionarily speaking, and it's sort of an open question that we don't have a lot of good evidence or answers for in the fossil record yet. It's always yet with the fossil record. You might find more fossils. You never know.
Yep. They're always there to be found, right? When you try to dig something up to build something, often you'll find a whole bunch of bones.
Yeah, one of the slothers that I know, that is her day job, is working for one of those environmental mitigation or environmental consulting or permitting, where they double check to make sure that we're not destroying our natural history heritage in the process of building new exciting infrastructure and whatever.
Yep. So let's get into your research. And now this is where you're going to be the best teacher ever, because I know you are. How you make the research of sloth teeth exciting. You gotta bring it home.
Okay. Well, my dissertation research that I did for my PhD wasn't strictly speaking about the teeth. I ended up using a lot of other tissues to sort of do my work. I shifted from my master's, which was focused exclusively on the teeth and looking at that microscopic surface texture. For my dissertation, I ended up looking more at the chemistry of tissues. Specifically, It's called geochemistry. So sort of an earth science perspective there. And
any
basically,
dissertation with feces in it is good.
Feces is good. And what I like to do, something I ask people to do on my podcast, so I'll ask it of myself right now, is to break down the title of a thing as clearly as possible, and I think that will help people understand the kind of work that I'm doing. So slow food movements is a joke, just because I was studying the diets of sloth and they are slow, and there is a thing called the slow food movement. If folks aren't aware of it, it's sort of an anti-fast food crusade campaign, whatever you want to call it. So we just thought that was funny, and my wife helped me come up with that, so a lot of credit to her. Exploring the dietary ecology, that just means looking at what animals eat. So ecology is the study of sort of how organisms interact within an ecosystem or within an environment. And so the dietary ecology is specifically, I like to say who's eating who. Sloths in this case are pretty exclusively folivorous in the modern world, which means they eat mostly leaves. In the past, we believe that their diet would have been, the ground sloth diets would have been more diverse. And so we I think that there's room to explore there. So I'm exploring the dietary ecology of extinct and extant sloths. Most people know the word extinct just means the species or taxa you're talking about is no longer living. Extant is the opposite of that, so that refers to the living sloths. Sloths, as we've been discussing, are a xenarthran mammal. Xenarthra is the group of mammals that contains armadillos, anteaters, and sloths. It's a pretty strange group of mammals that's endemic or evolved in South America, and have some characteristics that kind of make them the odd group out from a lot of other mammalian groups, and sort of they break a lot of the mammalian rules, so they're a fun group to study. Using stable isotope analysis, so that's sort of a big one to unpack. But basically, as most people probably know, when you look at the periodic table of elements, you get the weight of the atom. So you'll see that there's a carbon 12. Carbon has a weight of 12. That's the protons and the neutrons in the nucleus. And if you add neutrons, you don't change that it's carbon. It's still a carbon atom, but you change its weight. And when you add those neutrons to the nucleus, sometimes you can destabilize the nucleus. Sometimes you can make it unstable. So you see that in the case of something like carbon-14, which we use to do radioisotope dating. And that's because it decays predictably over time. But in some elements, when you add a neutron, it doesn't make the nucleus unstable. It just makes it a little heavier. So that's the case in like carbon. There's a 13 carbon. So most carbon out there is 12. A much smaller percentage of it is 13 and 14. The 13 is stable. The 14 is unstable. It's gotten too heavy. It can't hold together anymore. And so the way that ecosystems work is they sort of sort these heavier isotopes of various elements around based on the environment, based on the types of plants growing, based on the who's eating who, where things exist on the food chain. And so we can look at the ratio of the heavy isotope to the lighter isotope. We can actually learn something about the ecology of the source of that stable isotope ratio. So if we're looking at the food, we can learn something about where the sloth's food is coming from. If we're looking at the tissues of a sloth, we can learn something about how those foods get incorporated into those tissues. And if we look at the feces, we can see sort of the end product of that process. So one thing we like to say with stable isotope analysis is you literally are what you eat, and that applies down to the atomic level. And that's sort of what we're doing with sloths. And then when you can do that on the living animals, where you have a little bit more data about what it is they're actually doing with their time, with their life, you can then apply that to the fossil animals where you can't actually observe them. And sloths are a particularly good animal to do this with, because even the modern ones are pretty hard to observe and study. So there's a lot you can learn just by doing this technique to them in the first place. The slide, Scott, you've pulled up here is showing two of the systems I was interested in for my dissertation, the carbon on the left and the nitrogen on the right. So the carbon stable isotope value depends on the photosynthetic pathway of the plant. There's, roughly speaking, three main types of photosynthesis that plants do on planet Earth today. The main one is C3, which is over on the left-hand side, the left-hand side of the left graph. And it is the most common kind of photosynthesis that you see out there. It's also the oldest kind. Some newer kinds that have evolved to adapt to low carbon dioxide environments, not as much of an issue anymore. And aridity, which is regionally an issue, areas that are dry. They've evolved strategies to not lose water or not lose carbon dioxide when they are photosynthesizing. So we have the CAM pathway, which you tend to see in things like succulents, so cactus types of bromeliads, things that retain. Agave is a good example of a CAM plant. Things that retain a lot of moisture in their tissues. And then C4, which is also an arid, low CO2 adapted methodology for doing photosynthesis that you see in warm season grasses, shrubs like saltbush, and things like corn, which you might be more familiar with. One of the reasons I enjoy using wheat and agave and corn as examples is because those are agricultural crops that go into different kinds of alcohol. So you can actually use stable isotope analysis to see if your tequila has been adulterated with corn syrup or something like that, because it shouldn't show up as a C4 plant. It'd show up as pure CAM if it's good tequila. And then on the right side, we have the nitrogen pathway, and this tends to be an indicator of trophic webs or trophic level, and that's just a fancy way of saying food chain. That's how we tend to say it in ecology. That value tends to be the lowest at the primary producer level. So the image there is a Cecropia tree, which is one of the sloth's favorite trees to eat leaves from. And the heavier nitrogen if you think about the chemistry of it, even though it's only 1 neutron heavier than the lighter kind of nitrogen, when you're talking about, thousands, if not millions, of chemical reactions happening over the lifetime of an animal at the atomic level, that little bit of extra heaviness is actually going to cause it to sort differently than the lighter nitrogen. So as you move up the food chain, more and more of that heavy nitrogen kind of gets stuck in the tissues of the consumers that are eating the whatever's below them on the trophic level. So you're more likely to release a 14 nitrogen out into your urine or your feces simply because it's lighter. That chemical bond is easier to break. It's metabolically easier to move around. It might not sound like much on the scale of a neutron, but over time and on the scale we're talking about, it actually is enough that it can be measurable, which is really cool. A friend of mine made the point that, when we use this technique to study animals like dinosaurs or study animals like whales or study some of the biggest ground sloths that ever lived, which reached the size of modern elephants, we're taking some of the smallest parts of the universe and using them to understand some of the biggest creatures that have ever evolved. And I just think that's really kind of a poetic and beautiful way of thinking about it. And so with the nitrogen, you can see that, it's in the sloth than it is in the tree that it was feeding from. And then it's higher still in the harpy eagle that snatches the sloth out of the tree and carries it off to be eaten. So that's one of the ways we can use nitrogen to determine ecological dietary aspects of an animal's life.
Right. So that's I really like that kind of idea of that really small relating to something really big. That is, a cool thought. One of the other things I noticed when I was looking through these slides, your sense of humor comes out in the slides.
Oh, really? Oh, yeah. Thank you. Yes. The atomic
I was like, what kind of guy is Ryan? I'm looking through the slides. I'm like, okay, all right. He's a bit of a funny guy. I mean, in your dissertation, you put something like this in there. I mean, that's funny.
I mean, if you can't have fun with it, what's the point, right? And right. I think people retain information better when they're having a good time. I think I present it better when I'm having fun with it. So, just because the topic may be serious or have serious implications doesn't mean we can't have they're sloths. They're ridiculous animals. Like if you can't recognize the fun in that, I feel sorry for you, but I can, and I like to enjoy the sloths that I work with.
Yeah. So my one sloth story is I was in Monteverde in Costa Rica once, and I was on we're out walking around. This is a college trip, and our guide's like, oh, there's a sloth. And we're like, oh, finally we get to see a sloth. And he's like, it's way over there in that tree. And we're like, what? And then we get out the binoculars and we're like, okay. It was so far away. I got really excited. I'm like, oh, and I'm like, oh, it's just that blob over there, 500, meters away. I can't even see it, top of a tree. Okay.
Right. And my job is when that happens and I'm doing field work and working on the modern animals and not on the fossils, my job is then to grab the machete and be like, all right, well, let's see if we can bushwhack our way over there. Cause it's, it's probably not going to move for the next day or so. So you've got time to get over to it. Right.
Its species is probably still at the bottom of the tree. It's.
I love Monteverde. I've spent a bunch of time there and I still have some friends who live there and have great memories there. I always joke that if I die doing field work, it's going to be because I'm walking around a tropical rainforest where there are animals like fertile ants and bushmaster snakes, which are two of the most venomous snakes in Costa Rica. And I'm a person who doesn't believe that these animals are out to get you, but if I'm using these binoculars to stare at a sloth that's 500 meters away in the canopy, so I'm walking around the forest looking straight up, not paying attention to where my feet are going. I'm going to step on one of these snakes. It's going to bite me, and that's going to be the end. So, you'd think the risk from studying sloths would come from falling out of the tree when you climb up there to go get them, which is a risk, but a much less frequent risk than just encountering things on the ground where you're trying to get around this very dense forest to get as close to them as you need to actually do the study.
And I'd be more worried about, something like malaria,
I don't think that's a big issue. I think Monteverde, it gets too chilly for the malaria mosquitoes.
That's more like La Selva and down in the.
I've spent time at La Selva as well. That place, that's a very wet, warm area.
Oh yeah.
The area where I did my dissertation research is in Costa Rica. It's in the Limon province. It's at a place called the Sloth Sanctuary of Costa Rica. which folks might be familiar with if they've seen any of those like Meet the Sloths show on Animal Channel. And they're not necessarily a research facility. They're a ecotourism facility, but they were amazing to work with. They helped me out so much in getting access to these animals and let me stay, on site so I could be there, long enough to get all the work done. And so, I mean, and obviously, we had to work with the Costa Rican government to get the permits that we needed to bring these samples back from Costa Rica to the U.S. So I just have nothing but good things to say about Costa Rica and the Sloth Sanctuary and the folks who've helped me out and made this research possible along the way.
Awesome. Very good. It takes a lot of people to get research done. And
yes,
I don't think
I was actually
it's one of the things I try to press upon students. I tell them, that there's this myth that it's like, you're just one person in a lab and that's your research. I'm like, no, it's like it takes a lot of people to try to get good research done.
Yeah, I mean, it took a long time to get the permits from the Costa Rican government because they take things pretty seriously when it comes to like when it comes to an American saying, I want to come to your country, sample animals that are native to your country, and then take those samples out of your country, you got to really prove that what you're doing is worthwhile and valid and that you're an ethical person that's going to handle these things responsibly. And so I totally appreciate that it was a slow process. So, we jumped through all the hoops. We dotted every I, crossed every T. I had multiple talks with people because there's a series of treaties called the CITES treaties, CITES. It's an acronym that govern different endangered species and how they can move from country to country. And the way that the sloths are categorized is I didn't need any permission from organizations in the US to bring the samples in. I just needed to have all the permissions from Costa Rica to take them out. And I had all that, and I had them packed, in the way that other researchers had told me to pack them, as accessible as possible. I could show anyone at customs in moments exactly what I had and all the permits I had for it. And of course, when I got to the U.S. Customs, I pointed out, I clicked the button on the little kiosk that said I have animal products, and the guy was just like, what do you got? And I started to tell him and told him I had all the forms, and he just waved me through. And I was actually a little annoyed. I was like, I did all this work to get all these permits, and you don't even want to see them? You're just going to wave me through and let me go? I mean, I'm glad I got things, into the lab eventually, but it was like, oh man, that one moment where it all could have gone wrong, and I was like wishing for it to at least be a little more friction.
Right. All that time getting all that paperwork done. That's not the fun part.
No,
definitely not. So the other pieces, so I have these slides and we can just go, I don't want to go through all like 46 of them, but we can. Hey, Volcano Doc is rating with a party of 14. Welcome in. We are talking sloths with Ryan Haupt, who is here, the sloth expert, one of the only sloth experts I know. So it's probably only the ones you know as well. And I don't know anyone that doesn't like a sloth. They're like they're cute, right? Even in their own special way. And so we were just talking about how doing research in Costa Rica and the research he's doing for his dissertation. If you have questions, please put it in chat. Welcome in, and please, be here, hang out. And I am Scott, Science Streams, and Wednesday night we do chats. So I appreciate you being here. And Volcano Doc was on my show, I don't know, like a month ago. She was here chatting about her research that she's doing. So
she also gave me a lot of tips for how to do Twitch right. So I'm very grateful.
Oh, awesome. Glad you guys are connecting. So
apparently I have to fall out of my chair at some point, and
then it's all about how what kind of show you want if you're on stream. This is a pretty low key chat, so we just kind of, I don't want to say chill out completely, but so I'll put little shout out to Volcano Doc there. Great channel. Thanks for stopping in. And now the party's over. Since they're all here.
Someone asked where I am. I am in DC. We had We had some snow this morning, our first big snow of the year, and it sort of turned into a slushy, icy rain later in the day, but winter feels like it's finally arrived. So that's where I am in the world.
Well, we're getting our snow tonight, so.
Big storm coming in, but we're not here to talk about the weather. We're here to talk about an animal that would hate it if it was getting snowed on, and that is the sloth. Except for, some ground sloths that did live in colder places.
Well, to say, I think some of the sloths were colder weather during the Pleistocene, if I'm getting my geological...
That is one of the epochs in which sloths were alive. They go further back than the Pleistocene, but the Pleistocene is maybe the time period, at least in the paleological terms, that I am most familiar with. And it actually does seem like sloths spent a lot of time in caves, and going into caves might be an adaptation for tolerating colder weather. And so we tend to find certain kinds of sloths. Nothrotheriops is one that comes to mind, which is this iconic kind of southwestern sloth. You also see it called the Shasta ground sloth, and that's a sloth that you find in caves, and you find them associated with taxa that are still alive today that we know don't do well in the cold, like vampire bats. So if you find a sloth in a cave with vampire bats, you can sort of infer that, hey, maybe this sloth didn't like it when it was cold out, and it's going into these caves to stay warm, same as the vampire bat, which is kind of a cool association. So they definitely were able to tolerate places that were colder relative to sloths that are alive today, but they might have still been a little less cold adapted than some of like the big, woolly mammoths and bison that we think of as these kind of iconic mega herbivores that were walking around the environment 10,000 years ago.
Yep. And I know you answered this earlier, but why do you like sloths? Tropical Geek wants to know.
I will give a slightly different answer.
Okay.
But still a true one. I think of myself as a fairly energetic and extroverted person. This year has been tough. And I almost think that once I started studying sloths as my master's research, which was really just as sort of assigned by my advisor, but I found these animals really fascinating because we don't know so much about them. And a friend of mine who also works with the Sloth Sanctuary down in Costa Rica, her day job is working with dolphins. And we were talking once, and she was talking about how she loves her dolphins, but she really understands the sloths in the sort of way that they're slow and deliberate and not that social, and they just want to take a lot of naps and literally hang out. Like, it's an animal that literally hangs out most of the time. And that was she understood that in almost like a spiritual way. Meanwhile, we're at the aquarium, we're interacting with these dolphins, which are like dogs turned up to 12. And I'm like, I get these dolphins, man. I understand this hyper need to be, get attention and to play and to let's do a flip and let's talk about it. Let's talk to each other about it. And the dolphins were really clicking with me in that sense. And I almost wonder, do we end up spending more, as people who work with animals, do you almost find an animal that's your complement that you need to figure out because you don't inherently understand it? And so is that, for me, are sloths just weird in that they're weird mammals, but they're also weird because they're not they're not my personality, and vice versa with her and the dolphins. And so I think I like sloths because I don't understand them, and I want to understand these weird animals that aren't like me and are hard to understand for a lot of other reasons, but I want to know them so I can appreciate them even more.
Very good answer. You just find that you have a passion for something, right?
Mm-hmm.
And there's
exactly
nothing wrong with going with it, right?
Well, and I think once you start doing research, I think one of the hardest transitions in going from undergrad, where you're getting a degree by taking classes, maybe you do an honors thesis project or something like that, but I think one of the hardest transitions is figuring out how to actually do a research project and to ask those questions that can be answered through the process of research, given the tools that you have available at your institution or affiliated with your collaborators. And I found, and I think this is common for people in grad school, is once you kind of unlock that part of your brain that asks those questions and figure out the way that that's supposed to go, by the time you get to the end of a project, you may have an answer to the question you asked in the first place, if you're lucky. But you definitely have at least 3 more questions than you had at the start of the project. And so the questions tend to kind of just, they tend to just kind of fractal out and multiply infinitely. if you let them. And so then the hard part becomes narrowing your focus so you're not completely lost in the myriad questions you could be asking, and focus on the questions that you can actually answer in a reasonable amount of time with a reasonable amount of effort, and go from there. So that's one of the fun parts of doing research, is all the questions you get to ask and then figuring out which ones are worth answering.
Right. I often tell students that science isn't about finding answers, it's about asking better questions.
Mm-hmm.
Because if
and
you
a lot
do
of times,
good research, you ask better questions.
Yep. And a lot of times I found that, at the end of a project, one of those questions might be the same question, but now that I've tried to answer it once, I can answer it better if I tried it again, flesh out the data set, refine the methods, increase the sample size, that sort of thing. So.
Yep. So I brought up this slide with the graphic that you made. Includes feces, of course, so we have to talk about it. And what does it mean? Oh, I mean, what's happening?
Good question. So this method of stable isotope analysis, you are what you eat down to the atomic level. We have these machines called stable isotope mass spectrometers. I like to say that it's a big skee-ball machine for atoms, so it can sort them based on their weights, which again comes down to that, who has more neutrons than the others. And essentially the value, the stable isotope value of food is something you can measure by measuring the food that the animal is eating. And you can take the tissues that are produced from that food, and you can measure that as well. So you can measure the feces, you can measure the hair, you can measure the bone, you can measure the muscle, you can measure the keratin on the claws, which is like similar to our fingernails. And then you can think about like, okay, which of those tissues do we have available in the fossil record? Like, we have bones in the fossil record. In the sloth fossil record in particular, we have feces preserved in the fossil record. We have hair. We have fossil sloth hair. I'm not sure there are many paleontologists that can say that they've pet the fur of their study system in deep time, but I have. I've opened the museum drawer and I've pet the fossil sloth fur. And the problem is we don't know the value of the food that was going into the extinct sloth that produced the tissue from that food. And so one of the open questions when it comes to sloth ecology is, how is the food altered by the process of digestion, incorporation into the tissues? And I mean, it's funny because incorporation literally means to bring it into your body. So how does that alter the stable isotope ratios? What's the filter that the sloth metabolism is acting upon the food? And so the equation I have up here is showing that we can measure the tissue, we can measure the food for the modern sloths, and the thing we're missing is this thing called the offset. That's the amount that the isotope value gets filtered by as it goes from the stomach into either the feces or the other tissues that the sloth is making via its metabolism. And so part of my dissertation research was seeing, could we calculate this offset, this unknown value for modern sloths? And that's an offset value, or that's a value, that's a thing that's available for a lot of other mammal groups, but sloths are so weird that I just didn't trust the other mammal groups would be a good fit for sloths. So can we calculate that offset value for sloths and then take that and apply it to the fossil record and see if it makes for a different interpretation of the fossil stable isotope values that we can generate from the fossils we have available for sloths? Does that make sense?
Yeah, so basically it's a procedure that's been developed and used in other studies that you're going to apply, in this case, to modern day sloths, and then if it works there, apply it to the extinct sloths.
Right. And the key was we were pretty sure this technique would work, because it's if it looks to you like a very simple algebraic equation, that's kind of because it is. The real question was, were sloths going to be different than what we would expect for a mammalian herbivore, especially like a small-bodied mammalian herbivore with a pretty slow metabolism? Like, it's already kind of a weird situation to be in. And then there is a fair amount of stable isotope data for fossil sloths that had already been generated by other researchers, because this is a technique that's pretty common in paleontology. But if those data were being interpreted just using a mammal value, that's a typical large-bodied herbivore, is that a different interpretation? Do you then say that a ground sloth had a different diet than you would think if it was using than if you used a sloth specific value? And that was one of the big sort of big picture questions of my research: is how important is it to interpret ground sloth stabilized dope data using tree sloth offset values? I don't, again, I don't know that I came up with a definitive answer, but I came up with what I think is an answer, and hopefully a reasonable answer.
Right. And I think it's also important that, I like how you're not coming up with all this stuff yourself, right? Other people have kind of gone through this before, and I always try to tell students the same thing. You're not doing this by yourself. It's not like you have to reinvent something. You just take what people are already doing and ask new questions using the same techniques.
One thing I tell students and younger researchers, younger scientists, is literally, whatever field you're interested in working in, take some of the seminal papers, and better if they're more recent, or papers written by like the big names in the field, and read them, and then look for the phrase exactly or similar to, well, we assume that, blank, right? Or it's widely accepted that blank. And so like take those moments when you're reading those papers,
right?
Be like, that's the research question, because that's the thing that nobody's tested, because this person who is an expert in this field, they're one of the bigwigs, just said, we assume this is true. Well, in science, why assume when you can develop a hypothesis, and you can develop a question, you can develop an experiment and go do it. Right. So that's sort of the attitude I like to take.
And you know what assumptions do.
Exactly. Exactly.
Yeah. And that's a great point to talk about, looking at papers and seeing where that is. Because so many times you kind of get in that rut. Oh, we've always assumed this. It happens again and again in science. And then they go back and they look at it like, well, who said this initially? Oh, some guy in 1845. Right. Said this initially. It's like
well, this guy said that because he was mad at Thomas Jefferson, and it was this whole thing. And it's like, well, that's not, we things have changed since then.
Right. We've moved on.
Hopefully. Yeah. And so with the work that I was doing for my dissertation, it wasn't even sort of the bigwig in the field saying we assume. It was all these other paleontologists just saying, well, we don't have any other option but to interpret this ground sloth data using the values we have for the typical mammalian herbivore, which is usually an ungulate or a hooved mammal. So things like, deer, cows, antelopes, things like that. And so they didn't have any other value to use. So it wasn't, wasn't an unreasonable assumption. I'm not trying to like cast aspersions or throw shade at anybody. I was just saying like, well, it's still an assumption, and it is one that we can test. And so let's see if we can test it and if we get something different.
Yep. Sounds reasonable.
That was the hope.
Right. And
we'll see what blowback I get when I try to publish all this.
I know. Good healthy debate is fine, right? Because then you just go out and you get more data, right?
Yes, but I've seen and known many scientists. It's easy to say, oh, it's good to be wrong in science. It's good to have your assumptions challenged. It's good to have that good healthy debate. But like, humans are still humans, and we have weird egos and hangups and bugaboos, and it seldom, much like pure theory and versus actual practice, it seldom works out exactly how one would hope when humans are in the mix.
Right. And I imagine it's also when you're working with extinct species and you're trying to go from the living to the extinct, it's kind of hard to do that, isn't it? I mean, you're kind of like in two different worlds in a way.
Yes, yes and no. I think for folks out there who study geology, you have to develop what is called a deep time perspective. You have to think on the temporal scale that most humans are never trained to think about. Suddenly thousands of years becomes a pretty short amount of time. And so I try to maintain that kind of perspective even when I'm doing the modern ecology. The modern ecology, the best applications I have for the work that I'm doing would be conservation of modern sloths. I often will joke that there are far more extinct species on Earth than have ever been alive today. And so we don't, as paleontologists, we don't need any more extinct species. We're good. Like, if we could make things not go extinct for a while, I would be fine with that. Please don't add more on my workload. And so I try to think in terms of deep time. What is the grand story to be told about sloths, and what can we say about them going forward that can help keep them alive into the future, so that they don't become purely a part of paleontology, and that they maintain some of this modern biology and ecology that we find so fascinating and endearing today. And so I don't know if it's a perfect answer to your question, but I guess I just try to think, I'm trying to think in terms of what are sloths doing 1,000 years from now, as much as I'm trying to think of what are they doing 10,000 years ago, and hopefully that kind of keeps the perspective of connecting them to their fossil ancestors a little more concrete, a little less abstract.
Right. So it's kind of like, it's not that they're like, I phrase it as, I think, two separate worlds, when in reality it is the same world. You just have to think deep time. So
yeah, it's a continuum. Right. And it's important for me as a researcher to think about, like, the fact that even though ground sloths seem like these really weird animals that don't really have any sort of modern equivalent, like, there's nothing on the landscape today that quite functions. Just sort of what that slide is illustrating, is that sort of disconnect between the modern sloths and the fossil sloths.
Yep.
But it's important to remember that human beings did interact with these animals. Like, when Paleo-Indians and the first peoples arrived in what are now the Americas, they weren't called the Americas when they arrived, they would have been interacting with these animals. We have footprints in New Mexico where the footprint of a Nothrotheriops is inlaid with the footprint of a human, and you can only form footprint fossils within a matter of hours. So we know that these animals, humans also being animals, were walking in the footsteps of these sloths in their same environment at the same time. There was a fossil find, or I guess not a fossil find, a archaeological find in Colombia that was just making the rounds in the press recently, that appears to be the first cave art or paleo art of a ground sloth, probably mom and her cub, if you believe the interpretation. So to me, it's really fascinating to think about the fact that, like, the history of these really strange animals that not a lot of people have had the chance to interact with directly is also the history of us, and we were all interacting together at one point. And that's been lost to an extent, but it's not completely gone, and we can still see evidence of it in these really cool, amazing finds that are coming out literally every year.
There are fighting words in chat. The sapphire,
I see the sloths are yuck. They're not the cleanest animal for an animal that lives in the rainforest where they get rained on all the time. I remember I was spending a day working with some sloths, and I was just covered in dirt by the end of the day, and I was like, how does an animal that is never actually on the ground getting me covered in dirt? Like, that shouldn't be possible. But they can be quite dirty animals, but they're covered in, when the wet season is there, they're covered in algae. They've got algae that grows in their fur that gives them kind of a nice sort of plant smell, as opposed to like sort of that animal smell. They are covered in moths, so if you're not a person who likes bugs, I could see that being a little yucky. In general, they're pretty inoffensive animals, though. I've got a couple of shirts that were torn just because they were trying to grab onto me in weird ways that clawed at my shirt. But I would disagree that they are yuck, but I understand the perspective.
Now, is some of that, like, the algae and the moths, is that some of that symbiosis, or?
You asked me about debates before, and that is one of them.
Okay.
My answer would be it is as yet unclear what exactly the role of the algae is. There are those who think that the algae is a food source for the sloths. There's those that think the algae is a food source for the moths. There's those that think that the algae is there for camouflage. If you want to live in the trees and avoid predators, good. It is easier being green, regardless what Kermit says. It's a little tough to parse. It is one of those questions that remains, I think, unanswered. There's some tantalizing evidence. There's been DNA of the algae found in the stomach of some wild living sloths, so that would seem to suggest that they're consuming the algae to some extent. However, it's never been observed, so we've never seen a sloth licking its fur. They groom themselves with their claws. They've got these great claws. Why not, if you got an itch, scratch with your claws if you've got these great Wolverine-style claws. But then on the other hand, their hair that they have seems to be evolved with these like grooves in it. It's a great home for the algae to live in. And so there seems to be evidence on both sides of sort of this. Is it a symbiosis where the sloths are getting something out of that relationship, or is it sort of a non-parasitic symbiont that's getting a free ride in the sloth fur and it doesn't do the sloth no harm? So, what's the problem? So that is one of the big open questions, at least when it comes to modern sloths.
Yeah. And I think some of the Volcano Doc alluded to another, discussion that's always brought up with the megafauna in North and South America is the human impact, on those extinctions. So
yeah.
I mean, there's always that talk of, how much of that was environmental, how much of it was human, were humans just hunting them and driving them into pits and that was it? Or was it more subtle, where their effect on them? So that's another debate.
So that is a good question. It's a tricky one for sloths in particular, just because before, a couple of weeks ago, there wasn't a ton of examples. I don't think there were any examples of paleo art. So it was hard to know to what degree ancient peoples in the Americas interacted with these animals. If you think of paleo art as being like a sort of reflection of the values of a culture, if you're not painting something, then maybe it's not a valuable thing to you. Bison, if you've ever had bison, you might know that it tastes pretty good, at least in my opinion. And they live in these giant herds, so it's both tasty and lives in dense congregations that you can go out and hunt. The ground sloths may have been more social than modern sloths, but probably weren't living in giant herds and might not have been as tasty as a bison based on what we know about modern sloths. And they don't have a ton of fat, so they might not just have been very good to eat. That's all speculation. I will be quick to clarify. I can't say that with any certainty. So we have butchery sites for some of these large mega herbivores where we have fossils that have actual cut marks on them, right? Pretty clear evidence that humans were using tools to break down the carcasses to eat the meat. We don't really have that with sloths. We have a few sites that are maybe purported to be like that. And there is this big debate of, was it humans or was it environmental factors? So we're talking the end of the last ice age. This is 10 to 11,000 years ago. It is a period of intense climate change, much like today. It was a period of intense human expansion and exploitation of the environment, much like today. And then a bunch of things went extinct. But the things that went extinct tended to be the biggest animals on the environment. And that's kind of weird. That's not a type of extinction we see a lot in the fossil record. A lot of times when there's a mass extinction in the fossil record, when you think of the asteroid wiping out the dinosaurs or the end Permian extinction where 95% of life on Earth goes extinct, it's everywhere, right? It's on the continents, it's in the seas, nothing is safe, right? It's strange for an extinction just to target the biggest animals. It's not strange to think that humans would maybe go after the biggest animals in one sense, right? That's where the most food is. But it is strange because most of the time hunters don't go after the biggest animals in the herd because those are the most dangerous animals to mess with. So if you're a lion hunting an elephant, you're going after the baby elephant. You're not going after the giant bull male that's going to flip you into the river with the crocodiles. So there's factors at play here. I think you're going to have pressure on these animals from climate change, from the end of the last ice age, right? So it's going to be their environment was changing and they were stressed dealing with that. And then you add the stress of humans hunting them for the first time. And I think that 1-2 punch might have been enough to really knock them over the edge. An important thing to think about with large animals is they tend to have much longer generation time. So elephants have a gestational period of 18 months, I think. And they usually only give birth to one new baby elephant. And then it takes that baby elephant X many years to reach adulthood. And if you compare that to something like a rat or a possum that can have, a bunch of babies all at once, and they can do that every single year, and those babies are ready to go and reproduce themselves within a year or so. So it doesn't take a ton of pressure to really knock a large megafauna herbivore mammal's reproduction cycle out of whack. And if you knock it out of whack just enough at a period of intense climate change, that could be enough to create an extinction.
Yeah. I've also always had the hypothesis that when humans first, find a new animal, the first thing you do is you have to see if it tastes good or not. So that is an unfortunate thing for other animals. I mean, inquiring minds want to know, does it taste like chicken? You know, I mean, well, look at this new creature we just found. Wonder what it tastes like. Let's go kill it.
Some of my colleagues and I like to joke that, like, if hunting was a sort of thing of status, right? If you hunted stuff on the environment to be impressive to your community, it might just not have been that impressive to hunt a ground sloth. They might have just been too easy to track and kill because they were these big, slow, lumbering animals. So it just wasn't worth it from a pride or status point.
Yep. No, I want to get to one
again
graph.
pure speculation, but that's something we joke about.
So I was looking through your slides, and this graph here stood out where we have the food consumption and the temperature. And I'm looking at it, I'm like, that's crazy. That correlation is so spot on. Basically, you know if a sloth is eating or not, whether or not it's, you know, what the temperature is, right?
Yeah, it's interesting that you liked this slide, because this is one of, when I gave my presentation to my committee and to the public, this was a hidden slide. I didn't actually talk about this, so this is like exclusive content. This is based off of a study that me, my often frequent collaborator Rebecca Cliff, and some of our fellow collaborators did, where essentially sloths modern sloths. This is modern sloth data. So this is exactly what you said. This is temperature. So the three lines on the bottom are the average max temperature, the average median mean temperature, and the average low temperature over the course of a couple of months. And then the dotted line on the top is food consumption, average food consumption in dry weight food for a number of different sloths. And yeah, what we found was that when it was warmer, they were eating more. When it was cooler, they were eating less. And a couple of things to know about sloths is that they keep their stomach full pretty much all the time. So they don't really have, like I said, they don't have any fat. They essentially, because of where they live in the treetops of these neotropical forests, and they eat leaves. There's not a lot of vertebrates in these environments that also eat leaves, so there's functionally no competition except from invertebrates and insects and stuff. There's essentially a limitless food supply, so they don't really need to like store fat on their body for a rainy day, because a rainy day just means that there are enough leaves for them to go around and eat, and they're happy and they're fine. So they eat a ton of leaves. They keep their stomach full all the time. So the only time a sloth goes to eat new food is when its stomach empties out a little bit. And sorry, my dog is getting a little mouthy because I ran out of cheese to feed her. And a sloth's stomach is up to a third of its body weight, so that's like a huge amount of mass for this tiny animal climbing around the trees. And what we found is that when it was warm out, they ate more. When it was cold out, they ate less. And we think what's going on there is, if they're fermenting all of this stuff in their stomach, the fermentation, if it's happening faster when it's warm out, their stomach is emptying out more quickly, and they need to eat more to keep their stomachs full. And if that fermentation is slower when it's cool, they might not need to eat as much as often because their stomach isn't emptying out as quickly. And so even though they live in the tropics where the temperature is relatively warm year round, sloths, their temperature varies with their environment. So they tend to go up and down a lot, to the point where, like, I often say, if you were a human and you were experiencing this variation in temperature, like, we'd send you to the hospital. Like, there'd be something very wrong with you. For a sloth, that's normal, and it seems to affect their appetite. And I think it's the opposite from a lot of other mammals. I know when it's a really hot day out, I don't want to eat like a huge meal and be incredibly full all day. But if you're a sloth, that's what you do.
Yeah, I just thought that was great, and this is good. When I see it, I think, not higher ed. I think like pattern recognition here just for that graph. So
and we obviously,
what conclusions can you draw about sloths? It's like, well, you can look at it and be like, oh, duh, So, all right, here's what you talked about earlier, your sloth sanctuary.
That's Buttercup in her basket. She was the mascot of the sanctuary, and Pedro, the big ground sloth that greets you when you come in.
So your materials and your methods here. You weren't doing a tree boring with this. I assume that was for something else.
That was for the stomach, actually. we were working with sloths who were deceased. So they were living sloths in that they are the extant members of the group, not the extinct members of the group, but they had died, passed away at the sanctuary, which happens. They get these really injured animals brought in, and sometimes you do all you can for them, and it's still not enough. And they have a freezer, and they kept those animals in the freezer and allowed me to work on them when I came down to visit, which was very generous of them. And since I mentioned, this is a kind of a perfect segue, actually, because their stomach is, a third their body weight and they were frozen solid. We thought the easiest way to get stomach contents out of them might be using the tool you pointed out, which is an increment borer, which is used for taking cores out of trees. We thought, why not take the core out of a sloth's stomach? I'll tell you a story that didn't get included in my dissertation defense presentation, is that, again, these are gut fermenting animals. So there's gases being built up as they digest their food, methane and other things. And there was one sloth I was working on where I was having a hard time getting into the stomach using the increment borer. So it's got a, if you can't see it, it's very, at the very tip of it, it's got like kind of a drill bit, but it's a hollow tube. So whatever you drill into is getting sent up the hollow tube for you to extract and look at. In my case, isotopically, if you were looking at a tree, it would be counting the rings. And so I'm standing over this sloth. I'm giving it my all. I'm really cranking into it, trying to penetrate through the fur to get into the stomach. And I hit the stomach. This sloth must have continued to ferment after it had passed away and been put in the freezer. And so I'm standing over this open hole, and all the gas just shot up the increment borer right into my face. And it was one of the worst moments. It really made you question why you were bothering to do a PhD when you're breathing a face full of sloth stomach gas.
That's disgusting. Thanks for the image. That's the wonders of science right there.
Welcome to research.
Kids, if you want to do research, there you go.
I do not have video of this. I nearly did puke. This room we're in, the Sloth Sanctuary, has a building on their campus called the Slothpital. It is a sloth hospital where they do all the, clinical work on these animals, and they had a room in that Slothpital set aside for necropsies, which is what it's called when it's an animal. Not it's an autopsy if it's a human. It's a necropsy when it's an animal. So they had a room set aside to do the necropsies, and that's where I am in this photo with my Santa Cruz Go Slugs shirt on. And when the gas hit me in the face, there's a door to the outside, and I immediately went out that door and was, buckled over and trying to breathe deep the fresher air of the outside. And I was heaving, but I managed to not fully throw up. So it was close. We invested heavily in the Vicks Vapor Rub, and you put it, I didn't have a beard at the time because this is a COVID beard. And so you put it on the upper lip, so it's right above, right under your nose. So you're breathing the Vicks Vapor Rub instead. But it almost overpowered the Vicks. Didn't quite get me, but it was close.
Fun times.
Well, I mean, in hindsight, it's a good story.
Oh yeah. It's those things that you remember right there. So you got you cored a whole bunch of stuff. You clipped nails. You looked at bone. You got muscle. So all these different tissues, because you were looking at tissues, right? Yes. That was the intent. And then you're going to do the, isotopic analysis, on those tissues. Correct? Yep. See, I'm following. I got this. Right?
Yeah, I'm glad.
And we're looking at the carbon and the nitrogen, just like we saw in an earlier graph. If you weren't.
Yes. So. And typically you put the carbon on the x-axis and nitrogen on the y-axis. So that this is sort of the standard way to plot these sorts of data. I often like to point out, I try to be, whenever I'm presenting, I like to clearly explain my graphs every time, just because I think you can very quickly lose an audience if they don't know what they're looking at. So carbon on the x, nitrogen on the y. And the point of these two graphs is to show on the left-hand side, it's to show the tissues that grow continuously. So if you think about a sloth claw, much like our own fingernails, the portion at the very tip of your fingernail is the oldest part of your fingernail. It's the most distal portion, distant from your body, if you want to think of it that way. And then you have the proximal portion, so the part closest to your body, the newest grown part of the nail. And what we did on the left-hand side is we looked at hair and nails, because hair does the same thing. And we looked at the proximal and the distal tips from wild sloths that basically died right after ending up at the sanctuary, and then captive sloths that had lived at the sanctuary for a little while and been fed a consistent diet. And what we found is the green points on here are showing
Eureka!
that
Sorry, that was an event. Someone paid $800, so I had to scream Eureka because you found it.
Hey, Nikita, I was wearing a Santa Cruz shirt, and that's in California, so that's appropriate. And so the point of this graph on the left is just to show that the wild sloths are kind of all over the place. You can see that the arrows connecting the proximal and distal tips of tissues are not going in consistent directions. They're not going a consistent magnitude. They're not starting and stopping in the same place. But the brown sloths, surrounded by sort of the dotted ellipses, are the ones in captivity, and you can see that their data is much tighter. So they're basically, it's showing the effect of that controlled diet being provided by the sanctuary. And so when we calculated those offset values, the sort of missing unknown X in our equation from before, you can see on the right graph, the foods that they were being fed. So the sloths in captivity are fed the leaves from that Cecropia leaf, which is in the lower left corner of the graph on the right. And they're also fed some dog food because there's reports of these sloths eating animal protein in the wild. Those reports aren't super well substantiated, but it means that all the people that keep them in captivity give them some dogs. And the squares on the bottom of those arrows are showing what the stomach contents, the values of the stomach contents, and then the circles that they're connected to are showing the feces that came out the other end. And the point of this graph is to show that when you look at the captive sloths, even they start in a similar space, and the stomach to feces all kind of move up and to the right by about the same amount. And that shows that we were getting just good control on how the diet was affecting the feces that came out the other end. So when we calculated our offset values, that sort of unknown X that we're gunning for here, we only use the captive sloths just because that consistent diet seemed to be making a difference relative to sort of the chaos of the wild sloths.
Well, that kind of gives you a control in a way, so you don't have that variability.
Yeah.
Yeah. And it's kind of interesting, like, since you had some that did eat the dog food, you can kind of use that as couldn't you like look if you saw sloths maybe that are extinct that had more protein in their diet, you would see that as well, right? So you can kind of have a comparison.
It's sort of an interesting quirk that the feeding of dog food gave us an opportunity to look at that. I see a question about sloths mainly eating leaves. Yes, sloths amongst mammals are some of the most leaf-eating mammals there are. Three-fingered sloths eat probably 90 to 95% leafy browse. Two-fingered sloths maybe 85 to 90% leafy browse. Most mammals could not survive on a diet of just pure leaves in that way, but they've figured out a way to make it work.
I mean, who likes salad that much, really? I mean,
sloths.
Sloths. Man. But I'm sure that the leaves they eat aren't just iceberg lettuce either, you know.
No, they prefer the younger leaves from certain kinds of trees. They're very picky about the kinds of leaves they'll eat, and so each individual sloth, especially the three-fingered sloth, which is what's pictured there, will only eat the leaves from 5 or 6 different species of trees. So they're very picky. They learn their preferences from their mother. There's only a few tree species that overlap enough that you can even keep these animals alive in captivity. And they like to eat the younger leaves because younger leaves tend to have fewer secondary metabolites, which is just a fancy way of saying chemicals that aren't easy to digest. So younger leaves tend to be more digestible. Secondary metabolites are things that plants produce in part to prevent herbivores from eating them, but younger leaves are supposed to be the more productive leaves for the plant, so it doesn't necessarily put those secondary metabolites in there yet. But if you think about like that bitter taste in cilantro, that's a secondary metabolite. Like that's a chemical compound that the plant is producing to make it unpalatable for herbivores, and yet we humans have decided that we really like it. And so we use it in all kinds of tacos and curries and delicious things like that. I see another question. You want me to go ahead and answer that one, Scott?
Yep.
What adaptations do sloths' digestive system have to digest leaves? Time, is the main one. Time and mass, so they dedicate up to a third of their body weight on their stomach, so they just have a lot of room dedicated to holding as many leaves as they can, which is kind of rare for a mammal. And you have to remember, every movement a sloth makes, it's making it against gravity, right? Because they're these suspensory animals that live up in the trees. All of us kind of passively work against gravity just by how we stand on the ground as quadrupeds or bipeds, whereas sloths, they're hanging and climbing. So they have a very different sort of set of energistic sort of calculus they have to do every time they want to move around their environment. And so the thing that sloths have done is just to slow everything down. So the adaptation for sloths to digest leaves, which are very nutrient poor and calorically poor, is just taking a really long time to do it. Most mammals pass their food from consuming it through the gut out the other side in about 24 to 48 hours. So that's called gut retention time. Just think about that the next time you eat corn. You'll probably see the effect of that timing in person. And we find that amount of time is pretty consistent among mammals. So everything from primates that are a bit smaller than us to horses, which are quite a bit bigger than us, to elephants, which are way bigger than us, all have about a 24 to 48 hour gut retention time. That's about how long it takes the food to pass through start to finish. Sloths, some of them have been recorded as having gut retention times of up to 300 hours, which is 12 and a half days. So about 2 weeks. They only defecate. They only poop about once a week. So they just take a really to digest the leaves and to extract as much nutrient as possible, just by being really slow about it. So that is, you've sort of, and Burroughs, you've sort of hit on one of the quintessential slothiness of the sloth, right? They really are some of the slowest mammals out there.
Well, Moron has a good question. What about those really giant aquatic mammals? So what about like
really
the blue
hard
whale?
Really hard to study those. So that's one of those things where we're limited on. There's no blue whales in captivity. So how do you feed a blue whale a controlled diet that you can measure when it comes out on the other end? The way we do these gut retention time studies, there's a number of ways you can do them. You can do them, look using X-rays with sort of that milk of magnesia that is visible on the X-rays. Can't put a blue whale in an X-ray machine. You can do it by feeding an animal. The way it was done with sloths is you feed them a chemical marker, right? You find some chemical that is not toxic or dangerous to the sloth. You feed that with their food, and then you measure that chemical in the feces when it comes out the other end. And then you keep track of, okay, it's been 3 days. We've recovered 5% of the chemical that we introduced. It's been a week. We've recovered 50% of the chemical we've introduced. It's been 12 and a half days. We've now finally hit 100% of that chemical. So that's how long it takes the food to pass from the mouth all the way at the other end. Blue whales, you can't really control their diet. So it's a good question. There might be someone who does know a better answer than my non-answer, but off the top of my head, I don't know. I can tell you what is still a pretty big aquatic mammal is manatees. Manatees, excuse me, manatees that do live in captivity, so we can do this sort of work on them, are actually second place to sloths as far as I know in terms of gut retention time. And even then, they're at only about 150 hours. So they're about half a sloth, which is still way longer than a lot of other terrestrial mammals. But sloths are still king in that sense, as far as I know.
The non-answer was pretty interesting.
Hey, okay. Welcome to science.
I won't give you the answer, but I'll give you some of the other amazing facts. Awesome.
I got an argument online once with a guy who thought that this study that came out about blue whales didn't count because it wasn't an experiment. Like, this person just philosophically didn't believe that observational data about animals counted as real science. And I'm like, if you want to propose a way to do an experiment on a blue whale that's ethical and feasible and fundable, like, be my guest. But I'm not seeing that as a pathway towards a lot of productive science.
That's ridiculous. Like, oh yeah, it has to be a controlled experiment, and you have to have a population size of 10,000, or
Well, that's a frustrating thing with paleontology. Is a lot of times in paleontology, my dog has decided to join the party. She's finally stopped begging for treats and is willing to sit on the bed and chill. That's one of the the funny things about paleontology in particular is that, like, if you're working with a lot of modern biologists like I do, you'll show them a study of fossils that has a sample size of 12, and the biologist might be like, well, that's not a very big sample size, and you're like, well, that's literally all the fossils there are available of this thing, and we were doing a destructive analysis, so they weren't going to let us drill too much, and so we kind of had to work with what we had.
Yep. Oh yeah. So I was on this graph. Now we saw the one earlier. I can scroll up where it had your other results, and then we had this where we're actually looking at that feces and where the leaf contents were going, feces or into the tissues,
right,
using the analysis. And you got some results. It looks
like. So I mean, essentially this slide is just completing that equation that we had before. These results probably aren't going to mean much without me having gone through all the steps along the way, but it's functionally just showing that we were able to get an answer from the math that we did. I think the next slide is probably more informative for sort of some of the stuff we were talking about.
The A and B.
Oh, no, not this one. The next one. Yeah, I should know the order of my own slides, but it's been nearly 2 weeks since I...
The carbon and the nitrogen values amongst the different...
Yeah, so this is showing just sort of the main takeaway from this slide is just to show that, like, when you compare sloths to other kinds of mammals, they don't always overlap. If they do overlap, the sloths tend to overlap at sort of the extreme ends, one end to the other of the typical mammalian values. So, earlier on we were talking about one of the questions was how much do their offset, that unknown amount that the isotopes ratios change from the food to the tissues, how much does that change happen relative to other mammals? Here we're seeing that the change in sloths is different than what you would expect from other mammals on a similar diet or of a similar size or in a similar environment. And so it's showing that sloths functionally are doing something a little different based on a lot of probably, their metabolism and other weird life history characteristics of these animals. They just aren't quite falling where we would expect them or not expect them based on their weirdness to fall. And sort of that informs, again, that idea of how do we interpret the fossil data if we know that the modern sloths are quite different than the mammals that we have around today.
Right. And here's where the question too about the dietary ecology, the dog food versus the leaves.
Yep. This gets back to that idea that the captive sloths are being fed a diet that's supplemented with dog food, which is a little odd, but many herbivores will eat meat or animal protein when offered. That is a thing we see both in captivity and in nature. So sloths are no exception in that sense. And one thing you can look at, what this slide is showing, is that there's, you can measure the same isotope from multiple tissues or from multiple components of a given tissue. And so what this is showing is that when you look at the carbonate and the collagen, which are two of the components of bone, you can measure carbon from both of those tissue sources. And the difference between those carbon values is a pretty good indicator of the amount of animal protein consumed by the animal during the period of that bone's formation. And bone tends to form, it's constantly being reworked. So you're looking at the last few months to a year or so of an animal's life when you look at bone. And so if you look just at the terrestrial consumers, you can see that when you look at carnivores, so you see the little silhouette of the howling wolves, it's a lower space between those two carbon values than if you look at something like a moose, which is a herbivore, which has a larger space between those two values. And so really that's the quick and dirty way of thinking about it, is that, like, the more animal protein consumed, the closer those two numbers are to each other.
Right, and then we get to the discussion of food source, again, animal matter, and then
yep. And so just sort of this idea that the wild sloths, we think, were strict herbivores, even though the husbandry practices have sort of developed to reflect a potentially omnivorous tree sloth. So here we have an example where we had wild sloths that were...
Thank you for the bits, Nikita. So you give me some bits.
Nice.
Yeah.
So essentially, if you just look at the part over to the far right, you can see that the sloth expectations were that we'd have wild sloths that were strict herbivores and captive sloths that were potentially low meat omnivores based on this dog food consumption. And this would allow us to test to what extent meat consumption, animal protein consumption is evident in the stable isotope values of sloth tissues, which is a question that is relevant for the fossil record.
Yep. So we can actually draw some conclusions. That'd be nice.
All right. One would hope.
And then you try it out on your fossil sloths.
Yeah, this is not a study I did. This is a study that was done a couple of years ago by Bocarenz et al. And what they found is using fossil data, they were able to show that the ground sloths from this site in the late Pleistocene of Argentina, the ground sloths are matching our expectations for herbivores. But this result was challenged by some folks who have a hypothesis that sloths may have been cryptically scavenging or eating meat to some degree, even hunting. And it was challenged by them on the basis that, how well does this technique work for sloths? And that was something that we didn't know yet. And so my advisor Mark Clements and I realized that we had an opportunity to test this organically because these captive sloths that we were working with in the sloth sanctuary in Costa Rica were being fed a little bit of dog food. So we have omnivorous sloths that we can see. How much does this little bit of dog food that's being offered to them affect the big delta 13C value between the collagen and the carbonate? And if it shows up when you look at modern sloths, then it stands to reason that maybe it would also show up if you were looking at ancient fossil sloths. And so it would sort of hopefully bolster, I won't go as far as to say it's confirmed, but it would bolster the results presented in this paper that sloths were herbivores and not secret scavengers or omnivores or carnivores.
Yep. And this goes on to that same thing where you have
it's a hypothesis and we can test it.
Yep. So I'm going to go through these here fairly quickly. I don't want to be here till too late. I do have to wake up and teach tomorrow, by the way. It is a snow day, but it is a remote snow day tomorrow. Meaning,
oh, so lame! You should absolutely get snow days off, even in remote world.
But if we don't use snow days, I get the days off in the spring, which I would prefer because hopefully by then, I can actually go outside and do things.
So, well, if that's your preference, I understand, but there's something magical about a snow day.
There is.
An actual snow day.
So
this is that same data from the Bocarenz et al. 2017 paper. I've resorted it a little bit based on, so their data in that original figure was sorted based on taxonomic group, but also locality. I kind of got rid of the locality, and this is just based on taxonomic grouping and subdivided by diet if needed, and including my data from the captive and wild sloths. So what we're showing here is that, the carnivores, even the omnivores, are all plotting lower on this chart where we would expect them to. Some of the pure omnivores, like the rodents and the tesuids, which are peccaries, so like kind of New World pigs, are plotting right in the middle where we'd expect omnivores. We have our captive sloths, which are the green bar, which is dropped below that second dotted line. So those captive sloths, the ones that were eating a little bit of dog food, are plotting in more of the carnivore-omnivore space, whereas our wild sloths are plotting the median is above that second dotted line. Right. So they are plotting where we would expect to see the herbivores, and they're plotting just along with all the other herbivores, including the extinct sloths, which are the ones on the far end, the extinct xenarthrans. And so this is essentially showing that the sloths are meeting our expectations. When sloths eat a little bit of meat, we see it. When they're living in the wild and we think that they're not eating any meat, we don't see it. So this sort of, hopefully supports the claims being made by the previous researchers in suggesting that this technique on sloths is valid. It works, and it tells us what we would, in this one sense, sloths do follow the mammalian norms. And that was good. We were glad to see that.
Yeah, and I'm, and you would be looking at similar things with teeth as well, right? Because when you examine the teeth, of course, you're looking to see whether it was eating meat or vegetable. And that's what you can get to here. But you're not getting that fine of what kind of plant matter it's eating using, this kind of research.
Teeth are a little tricky, at least with the work I was doing with the dental microwear textures, because you have to think about like what are the material science properties of the food being chewed on. So something like bone is very brittle, but a nut, like if you're cracking into like the pit of a fruit, can also be very brittle. So you can form the same texture with meat and with plant matter. So you do need to kind of also incorporate some element of just being smart about, like, if you're looking at a carnivorous mammal, you need to look at a type of tooth that's going to be chewing either meat or bone. And then you have to make your interpretations based on, okay, well, this hyena probably wasn't eating coconuts. This is probably the texture of a bone that we're seeing, not the texture of like an avocado pit, if that makes sense.
Right. Okay, now we're looking at some background, the area where they lived.
This is Nothotheriops shastensis, that iconic southwestern ground sloth, and in C, that is a piece of poop. That is a 11 to 30,000 year old piece of poop, and this is the cave that poop was found in. The entrance to the cave is on the lower left, and then the one you were just on the topo map. It's a cave I've not actually been to. I would love to go at some point, but it's a cave that was explored by archaeologists and paleontologists back in the 1930s, and they found just loads and loads of sloth feces and some bones to go with it. I don't know if people can go into Gypsum Cave. I probably held by a mining corporation right now, but I think you can get permission to go in. So if somebody knows anyone who has permission to go in, please let me know.
Wow, look at all that poop. But
yeah, and the photo of all the poop is actually that's a photo from Rampart Cave, which is in the Grand Canyon. So that's controlled by the National Park Service. There's actually about half a dozen other fossil sites or cave sites in the U.S. that have fossil sloth poop in them. So it's, relatively rare as a type of fossil, but honestly, like, given, all the other amazing mega fauna that was living in North America at the time, the sloths are the one where we have this kind of data set with all this poop in it. So that's pretty cool. The way these are preserved is these are really dry, arid caves. So the sloth poops the poop, and then the cave environment just kind of sucks out all the moisture. And it's a fossil because it's old enough to be truly considered a fossil. So anything about 10,000 years old or older, we tend to consider a true fossil, but it's not lithified. It hasn't been like turned into stone. So I call them coprolites, which literally means poop stone, even though they have not been turned into stones, but I call them coprolites just because they are true fossils in that sense. So hopefully that's not confusing.
Yeah, so lots of questions about poop. I have a couple myself. Did they just, Volcano Doc, did they just poop in caves, or do caves just preserve coprolite poop better?
Probably both. Modern sloths tend to be pretty specific about which trees they poop at the base of. So modern sloths actually climb down out of the tree to defecate, and they probably do so because they have there's a lot of theories about why they do so. It's not totally clear. One of the thinkings might be that these latrine trees are sort of sloth message boards where they kind of leave all their chemical information for other sloths in the area to come sniff out and find. With the sloths in these cave environments, the amount of coprolite material in these caves is so extensive that the sloths must have been using these caves for long periods of times, potentially generationally. There's a theory that the sloths were using these caves to give birth because it was a safer, more secure environment. That's a hard one to test. So if anyone has any ideas for how to test that, I am open. It's possible, something we talked about earlier is that when it got climatically cooler out, sloths use these caves as thermal refuges. So caves tend to approximate the average temperature of an environment over the course of a year. So in a cooler environment, a cave is going to tend to be a little bit warmer. In a warm environment, cave's going to tend to be a little cooler. So it's possible the sloths were using these as a place to stay warm when it was a little cooler out, and they just happened to be pooping in there because that's where they were living. I don't believe any coprolites from sloths have ever been found outside of a cave environment, so I don't know how we would test that exactly. And then I think Volcano Dog also asked about how long it takes the sloth coprolites to lithify. As far as I know, we don't have any lithified sloth coprolites. The only coprolites we have are ones that have just been desiccated, and it's all original material other than the moisture. So that's kind of cool.
Wow. And I would think you would like poop in the furthest corner first and then fill in. But
they might have. I mean, there are like
and then they did it again.
Layers.
Okay, there's a lot of interesting research questions you could go into just in a cave of poop.
Oh yeah. There's layers upon layers of stuff. I mean, Gypsum Cave, the poop has been dated. As some of the poop is as old as 30,000 years. Some of it is as young as like 12,000 years. So I mean, that's a long time to be pooping in a cave, even for a big sloth.
Yep.
So this slide is cool. This is showing some of the isotopic data. The green dots are the modern sloth feces. The brown data points are from the coprolites. But the cool thing about this photo is the microscopic images that we took. In an image A, it's a little piece of twig, and that little sort of light green line on top of the twig in image A is actually a thorn that was still on the twig that had survived all the way through a sloth's digestive system and come out the other side, which sounds uncomfortable to me, but this sloth didn't seem to mind. Yeah, exactly. So that's wild to me. And then in image B, that's just a piece of leaf material that had been eaten. And what's interesting about that is most of these cell walls that you can see there are still intact, which means that they hadn't been, if the cell walls are intact, then it really hadn't been digested that well. So it's suggesting that something about the digestive efficiency of these animals might have been quite a bit lower than the sloths we think of today, as taking a really long time to digest their food as completely as possible. So I was kind of looked at it under the microscope.
Yep. Well, I'm reminded of I've heard that like deer are that way, where they've found deer have starved to death but they have full stomachs because their diet was just so poor and their digestion so inefficient. So
sloths are almost the opposite, where it's like they're I like to say that sloths are constantly living in an modern sloths at least are living in an all-you-can-eat buffet, and they're almost always starving to death because they have no fat on their body, so they have no like reserves to pull from. They just eat whatever they can. These are pollen results, so we pulled some pollen grains from one of the coprolites and counted the grains, and it's interesting to see the sort of discrepancies here because how does pollen get into a coprolite? Well, it either blew into the cave on the wind after the coprolite had been deposited, or was eaten by the animal and passed through the digestive system along with everything else. We're kind of leaning on sort of the latter explanation here, but then you have to ask the question, okay, does the pollen represent plants that the animal was eating? It would have had to have eaten those plants in the spring when they were producing pollen and eaten the flowering bud, or in a desert environment like the American Southwest, is pollen flying around because a lot of it's wind dispersed, and so our plant's just covered in pollen, and the sloth is eating the plant that it wants to eat, but it's incorporating or ingesting the pollen from the plants nearby and in the environment. So this is sort of just looking at, what do we think? Do we think, this pollen data represents the environment at large, just all the pollen that was floating around, or does it actually represent the sloth's diet? That's still sort of an open question, but it was sort of an interesting way to compare to the isotope data that we generated.
There's that thorn again. You want to talk about this one? Or are we going to go on?
That was one of my hidden slides. We did, isotope data from all of the different component parts of the coprolite that we could take apart. But I don't find this chart, unfortunately, particularly informative. So yeah, we can move on. I wish it was more informative. I wish there was a clearer signal in the noise there. And this is sort of my bringing it all together for the coprolite study, and it was taking the stable isotope values generated from other sloth fossils and other studies, and we were taking it, we were correcting it all back to the diet based on that offset calculation we made with the modern sloths. And essentially what we're seeing here, and I think it's on the left side of the slide, all the data on the left-hand side are all the other sloths that have been sampled previously. All the data on the right-hand side are the data from the coprolites of the Nothotheriops, and showing that this desert sloth seemed to truly have a different lifestyle, different dietary preference than the sloths living in maybe more temperate forests with more rainfall and lower temperatures. On the right-hand side, we're seeing on the very top, the top row, We have the brown box plot and the gray box plot, and that's just uncorrected data from coprolites and from bones and teeth. And then the middle section is correcting those data using mammal offsets, and on the bottom is that same data corrected using sloth offsets. And the point there is to show that when you correct using one or the other, you get a statistically significant shift in your interpretation of that animal's diet. So if you just corrected it using the mammal offset, you would interpret an animal eating predominantly one kind of vegetation. If you correct it using the sloth specific offsets, you get an animal eating predominantly a different kind of vegetation. And when you use the sloth specific one, the Nothrotheriops, the Shasta ground sloth, more or less shows up more like a desert or dry climate adapted animal. So that maybe suggests that the sloth specific offset is a little better for interpreting the dietary ecology of an extinct sloth. I like to say that I'm not arguing that it's better, but it's different, and it's different in a statistically meaningful way. And so it requires us to keep thinking about it and keep asking these questions and exploring what it might mean given, how we interpret these sorts of fossil data moving forward.
Questions, answers, conclusions. Bringing it all home.
The conclusion slide, I often just like to say that the way I think about my work is, this sort of sloth Ouroboros where we have questions posed by the ground sloths that we can maybe answer using techniques from paleontology but applied to modern sloths. And then we can take the data we learned from those techniques applied to modern sloths and reapply them or apply them in different ways to the ground sloths to potentially then learn more about what the ground sloths are doing. And so I like to think of it as, one type of research feeds into the other type of research, and it just kind of becomes a cool cycle where both living and extinct sloths can help refine and help us learn more about each other as we, move ahead. So that's sort of my big picture take home message. And that sloths are weird. That's my other big message.
It's also weird. All right. Well, that was, I know more about sloth and sloth like physiology and evolution than I ever had before in my life. And that goes for everyone else in the stream. So, but it's a really good study. And a great example of, science in action. And that's what I like to see. So many different ways to look at it. Now, the last thing I was going to talk to you about, and I told you I would ask you about it, is you were involved with Marvel at one point. You were like the science advisor on their blog or something. Is that what it was?
Yeah, sort of. When this is probably 10 years ago at this point, it was when Iron Man 2 was coming out, was the impetus behind it all. And one of the guys who was in charge of finding and producing content for Marvel Digital, Ryan Panagos, who's still a friend of mine, knew through some mutual contacts that I was something of a science guy, but also something of a comics nerd. And he was like, "Hey, would you be interested in redox conditions of Iron Man?" I actually didn't do any geology of Iron Man, which would have been a really fun angle on it, but I didn't think about that at the time. They wanted some sort of science of Iron Man content for the website. And the funny thing is, the Marvel movies actually have science advisors. So there's a group called the Science and Entertainment Exchange, which is run by the National Academies, and its job is to connect scientists to storytellers of all types, but also storytellers in Hollywood at some of the highest levels like Marvel and Disney, and help them not necessarily get the science right, but maybe get the science reasonable if at all possible, or at least find plausible explanations for things. And the crazy thing is, I wasn't connected to any of that. I was just told, Iron Man 2 is coming out. Take what you saw in Iron Man 1, and if you can come up with a series of articles about the science of Iron Man, we'll publish those, and we'll use those as some science of Iron Man. I did one for Thor as well before Thor: The Dark World came out, and it's funny because when you talk to engineers, when you talk to physicists, when you talk to people who know about this stuff, I'm not an expert at the types of science that go into Iron Man and Thor. I I wrote these articles, and I wrote one about, how does the armor work? I wrote one about how does the user interface work, because we don't have like these kinds of suits that exist in reality. How does flight work? If he doesn't have a huge tank of fuel on his back, so how is he flying around? How do his weapons work? With Thor, I wrote, how is Thor moving around between worlds? And like, some of the feedback I was getting from the editors that I was working with to produce these articles was like, so you without meaning to, by just using the science that and based on the science that we're pulling to make these movies, you're kind of spoiling some stuff that comes up. So we're gonna have to edit these to like remove stuff that you just inherently predicted. Yeah, by getting the science right. I think for me, the biggest takeaway that I have from the Iron Man articles in particular is that I think in reality, even no matter how it's depicted on film, I think the Iron Man suit is probably at least 7.5 feet tall and much bigger much bigger than it actually seems. Because I think Tony Stark actually needs a lot more room inside that to cushion himself against all the impacts he's experiencing when he gets knocked around by Hulk and Thor and Thanos and all them. So, yeah, it was really fun. And then my buddies who run the website iFanboy, and for a while that website had a lot of written content. It's now just a podcast. They had me writing sort of a Science of Comics column every week, and so I've spent a lot of time writing and thinking about like, how does the science of comic books actually work, and what does that mean, or how does that how do the science of comics actually influence real science? My undergraduate advisor will say openly that his sort of inspiration to become a scientist was Professor X. He was reading X-Men comics as a kid and was like, well, I want a team of mutants to go, fight battles for me and do my bidding. I better become a scientist so I can figure out how to do that. So, I think there's a lot of fun to be had at the intersection of pop culture and science, right, especially sort of sci-fi pop culture.
And I think especially when you're younger and you get this thought in your head that, okay, I'm going to go do it. I'm going to become Professor X. It might change, but that seed was planted somewhere in your head, right? It's still there, and developing, and it needs to, even if it's nourished and it's fed well, then, you become a scientist. And as a teacher, those are the types of things that I try to get kids to see and experience so they can get that seed and they can get nourished. But also I try to tell them, like, there is so much more to science than just what you might think. That's a perfect example of science of comic books. It's like, it's a thing, they have science advisors for, movies and TV shows. It's, it's not just you sitting in a laboratory, which is what they think. There's a lot more to it. And you never know, like your experience, you never know where your path is going to take you. It could be 10 years. You could be, somewhere else, still doing sloths maybe, but, advising something else or doing something else. So I find those stories interesting, because it shows where you can put your passions in with your work, and then your work with your passions, and sometimes they come together and it's beautiful. So
I've never gotten to work on a Marvel movie, but because I've made friends in the comic book industry and in other similarly related industries, I do get people reaching out to me saying like, hey, I've got this like kind of kooky sci-fi idea. It may not work perfectly, but can you give me any sort of thoughts on how to make the most sense of it? And one time I was at a comic book convention back when we were allowed to get together in person in groups of several thousands. I was hanging out with a buddy of mine who's a comic book writer, and James Kalikios, I think I'm saying his last name correctly, who wrote the Physics of Superheroes textbook, happened to come up to his table where he was tabling and signing books and talking to folks. And they got to talking, and James, or Dr. Kalikios, Jim, I think he goes by, was like, hey, well, if you ever need any science consultation, with any of your comics you're working on, here's my card. I was like, hey, don't, it's my territory, back off. I got the science consultation for this guy. So it's
funny.
You never know where it's going to take you.
Oh yeah. Well, it was a great conversation tonight. Loved hanging out with you and talking, and hope everyone else here, enjoyed all this information. And it was great. And hopefully you had a good time, and you're more experience doing some streaming chats, because I know you're a big podcaster, and definitely check out his podcast. I'll put your information out there again.
Thank you. Yeah, my podcast is called Science Sort of. It's actually quite similar to this. It's a very conversational style show where, you know, folks who enjoy similar topics but don't always know about the same things will get together and talk about our science. And we call it Science Sort of just because it could go off the rails, and we could end up talking about. We did an entire episode once on the mechanics of R2-D2's flight in the prequels and how it doesn't make any sense because he's not aerodynamic at all. So, you know, I think it would fit in well for fans of this channel to check out my show, Science Sort of, which is at sciencesort.com and wherever you get your podcasts.
Yep. Well, awesome. Thanks for being here. Right now, we are going to go on a raid. So we're going to go visit another channel, and we're going to go visit OpenSet, who I believe is doing some microscopy while doing some synthesizer music at the same time. So if please hang out for a little bit, say hi to over there, and say goodbye to Ryan, and take care. We'll be in touch. Thanks for being here. And
yeah, thanks for having me.
Go.
Bye, everybody.
And we got 6, 7, 8 seconds until the raid, and awesome. And let me check and make sure it went through.
If you can hear me yet, how's your stream today?